Hi, I would like to take a few minutes to just introduce you to some of the ideas in synthetic biology, try to distinguish them from things that we do with traditional genetic engineering, and also just hint at some of the things that can be done with the BioBuilder program. The way I'll start is by talking about a common problem, or at least common for me, and that is that although sometimes I can grow beautiful plants like what you see on the left here, this was an avocado plant that my son and I grew from a seed of an avocado um, that we bought from the grocery store, and it was doing great. And then off we went on a vacation, and this plant, as well as many others, we return and they are shriveled up because they have not been watered. So there are solutions that we could come up with for the challenge of keeping plants alive while we are not at home. If I were an electrical engineer, which I am not, I might be able to rig up something that looked a little bit like this. It has a breadboard with particular parts associated with it, and this is from Make Magazine. I can even get a plan for how to connect those different sensors and resistors to the breadboard in order to have this electronic solution know when the plants were getting dry and know when to water them. An alternative would be from a mechanical engineer's point of view, and again, I'm not a mechanical engineer, but this is a pretty clever one where the water at the bottom of the pot holds the pot upright, and so when there is sufficient water in the plant, it holds the plant straight up and down, but then as the water in the pot starts to diminish and the plant gets dried out, it starts to tip over. And maybe this wouldn't help while I was away on vacation, but I could definitely, during my day-to-day, be able to see when my plants were starting to tip over and remember to water them. So instead, because I'm a biological engineer, I can think about how to perhaps solve this challenge of keeping plants alive and watered and green using biological engineering techniques. So one possibility would be to take, and this is a fanciful possibility, but you take a gene from some organism that is desiccation resistant, something that doesn't dry out too easily, like a camel. And if we knew what gene allowed that camel to resist drying out, then maybe we could just take that piece of DNA and drop it right on into the plant, and then the plant wouldn't dry out quite so much. And while we're at it and while we're dreaming up this system, maybe we also want to take a gene from an organism that changes color so that we could know that the plant was starting to get dry because it would change color. So maybe we can take a color changing gene from like a chameleon and drop that right on into the plant. So it sounds a little crazy, and I actually don't think this experiment would work, but the techniques for thinking about it and for perhaps even piloting it exist. And those are the techniques that we usually think of as genetic engineering. So the ability to amplify sequences from a genome, that's with polymerase chain reaction or PCR, so we can get many copies of the genes that we're interested in. We can identify which genes we're interested in based on sequence analysis, so the ability to read DNA is very important for genetic engineering so that we know what genes we have. And then finally, recombinant DNA, or rDNA, is the ability to mix and match pieces of DNA using restriction enzymes so we can patch them together in a way that we would like. So this is perhaps possible, as I say, maybe worth piloting or trying in some form. But what if we wanted to do something really fancy, like what's shown here? If we wanted not to just have a plant that would change color and maybe not dry out, but rather have a plant that we could take as a seed and plant in the ground, and over time, maybe not even too long, that plant would grow, and it would grow into a two-bedroom, two-bathroom house with a walkout porch. And this is a dream project from a group called Terraform, and their idea is a little bit different. They actually would like to use plant materials to fabricate walls and things that could then be assembled into this fancy house that grows from a seed. But with the field of synthetic biology, maybe we can dream about how to build this house from a seed using genetics. So Clearly, the techniques that we just talked about, recombinant DNA, sequence analysis, and polymerase chain reaction, are going to be insufficient if we want to try something as grand and as fanciful as growing our own house. And the tools of synthetic biology are added to these tools of genetic engineering with hopes that we might be able to someday design in a rational and robust way these complex genetic systems, like the tree the house that grows from a seed. And so there are three techniques that I want to just briefly introduce. The idea of standardization of parts, abstraction, and synthesis. So synthesis is sort of the simplest, and I'll start there. Synthesis is simply the writing of DNA. So just the way sequence analysis is the reading of genomes that exist. And so on the left of this slide, we have our chameleon, and it has DNA within it. That's a physical material. Through sequence analysis, we can turn that physical material into information, digital G's, A's, T's, and C's, and know which genes are within that chameleon. And then through DNA synthesis, this next additional tool, we can take that digital information and make it physical material again. There are compounds that can simply be added together, and we can write DNA. I will mention two quick things that are worth exploring further as you delve into synthetic biology. First is that the synthesis of DNA is getting less and less expensive all the time. And so it gets easier to do, and easier to do by more people who could perhaps afford it. The other thing that I'll note is that not everyone likes this idea that we can write DNA code. Some people feel like we don't really know what we're doing and can't know what to say. Some people feel that it's just not something that people should do at all. And these are really important and complex issues that are worth thinking about and discussing as you consider doing some of this work. So moving on, the next sort of tool of synthetic biology that I want to mention is standardization. And you may be familiar with standardization already in many ways, actually. If you think about many of the things you use day to day, there are standardized plugs and outlets and different ports that allow parts to fit together. But in terms of education, one of the most powerful educational tools that uses standardization is the FIRST robotics competition. This is a slide from an older competition, but it's just here to remind you that many teams and many people get very excited about building things from standard parts. In the FIRST robotics competition, teams are provided with a common kit of parts and are challenged to carry out a particular mission. And what's interesting, of course, about the FIRST robotics competition is that even with that standard kit of parts, they get many, many different implementations and ideas around how to carry out that mission. There is a competition in synthetic biology called the International Genetically Engineered Machines Competition. It's spoken of as iGEM. And that one is similar to FIRST robotics in that it asks the question, can you build a biological system from standard interchangeable parts and have that operate in a living cell? This has been it's been initiated in the collegiate division, so colleges take time over the summer. Teams are working with this kit of standard biological parts to build living machines. There is a high school division as well as an entrepreneurial division that is popping up as well, with this idea of maybe building from standard parts complex genetic machines. And then quickly just touch on this notion of abstraction, this idea that we can take complex ideas and perhaps not understand every bit of the complexity, or not focus on every bit of the complexity, but rather black box those complex ideas and allow us to understand and use them rather than try to understand every bit about them. And so as an example, I just want to use one of the iGEM projects that is an interesting one, in which a team of MIT students spent a summer to make smelly bacteria more pleasant smelling. So they allowed bacteria that normally smell stinky to become banana smelling at a certain stage of growth. And as a student of mine was watching the video of about this team project, That student wrote a comment on our class site which said that video was more than halfway over by the time I realized that BGD and WGD were actually complexes of genes, promoters, inverters, and other pieces of DNA that when coupled together could synthesize the chemicals needed to produce the banana and wintergreen smells. In explaining their project, the iGEM team had essentially reduced the complex process of biological manipulation to seemingly simple matter of starting with some input, precursors, that would produce a desired output, that is a smell. So I think this really captures this notion of abstraction and black boxing. Certainly, we know and can understand how the input, isoamyl alcohol, becomes the banana smell, isoamyl acetate. But to use that part in a complex system, maybe we don't want to be thinking about all those details all the time, and we'll simply black box it and turn it into a banana generating device. And so that is a quick introduction to synthetic biology, a distinction from genetic engineering. And I'll just mention that the banana smell generating bacteria, as well as other synthetic biology projects, can be investigated using the BioBuilder content. It has animations that introduce these ideas and challenges, several hands-on activities that you can do in the lab or in the classroom, and ways to study this emerging field of synthetic biology. I hope this has been helpful. I hope that you'll continue to explore synthetic biology as a field, and good luck and have fun.